It's a great pleasure to be invited to the DataFest. Probably it's my third time on DataFest, and I remember Facebook just reminded me that 4 years ago I was presenting in Armenian. And why my talk is about DNA models? I think that actually right now large language models, they produced very big impact on everyday life of everyone, but also the next big wave of impactful applications lies in biology and in medicine. And of course it's nice to have something which will help to create new medicines, drugs, and help people to live better, healthier lives. But also I think that very important applications is applying all these AI models to biological discovery, because this will help us to address very deepest question of science: origin of life. Can we have different life from ours? And what is happening during evolution? So this is why I found this area very fascinating. And it's also, it's not only impactful, it seems to be very promising. Why? Because we already know that AI models can learn language from sequences of words. So we have very simple task, just next word prediction can allow model to develop very deep understanding of natural language. But on the other hand, evolution created very nice language-like information storage or description of organisms as a DNA molecules, which is a foundation of our genome. So we have sequences in natural language and we have sequences in our genome. So why not apply the same approach to language of life? And this is basically very simple idea, and what we did, we just applied the same techniques to DNA, and today I will present what we have found. Is it applicable or not? But before we start, just a short intro into genomics, and I will consider genome as a program for organisms. So it's like some program which allows you to run all the processes and build your organism. And this is a very nice metaphor, and it shows you that you can directly apply all the current methods to genomes. So as you know, in every cell of your organism you have a nucleus, and in nucleus you have very, very long molecules. So in every nucleus you have DNA molecule, it's 2 meters long. So in every cell, 2 meters long molecule. And this molecule has like particular structure with where you can find 4 repeating parts, which is designated by T, A, C, and G. It's residues which like build this very long molecule. And we can say that these 4 base pairs, it's like nucleotides. It's a letters of our alphabet of language of life. And on the other level of complexity, we unite these letters by 3 to form so-called codons. And codons, they code for specific amino acids or for specific control signals for reading out DNA. So we have pretty strict genetic code which describes our organisms, organisms for every living creature. And actually every organism on Earth shares this genome, this genetic code. So the genetic code is the same for all living creatures we know now. And how it works: so we have this very long DNA molecule, and we have some parts of this DNA molecule which scientists, which biologists call genes, and usually this gene contains instruction to build proteins, and this is what AlphaFold is about, like predicting this 3D structure of these proteins. And this determines function of the cell, inside the cell for specific gene. And this is called gene expression. So it's like building blocks of our organism, and all the biological reactions, they are mediated via proteins. On the other hand, we have so-called here non-coding RNA, and these parts are involved in gene regulation. So we have gene expression creating proteins, and we have non-coding RNA expression, which is involved in regulation of genes, because every type of the cell in our organism has specific pattern of gene expression. So your blood cells or your muscle cells, they have different proteins expressed. So they have different phenotype and different function in our bodies. And of course, what is fascinating, that if you have this instruction, if you have the same instruction, you can create two very similar human beings. So these are twins which has the same DNA, and you see that they have very, very similar phenotype, and usually they have not only similar like externalities, how they look like, but also usually they have very similar behavior, character, and this is fascinating that we have such like pretty deterministic source of information about our phenotypes, and it gives us very big promise that if we will find a way to decode this information, so potentially if we have strong model, we can predict full phenotype from this information. So because nature already is doing this. So what is happening today in application of AI in genomics? So actually, if we look at the history of natural language processing, you can see that about 10 years ago, we have a bunch of specialized models, some model for question answering, some model for name identity recognition, and other for sentiment analysis. And about 2020, we have seen rise of transfer learning, when we have trained some language model which has pretty good representation of language, and after that, we fine-tune it for specific tasks. And the quality for solving these tasks, the same tasks, became much better, because here the model has some a priori language learned during pre-training how the language works. And of course now we have LLMs which can solve a lot of this problem even without fine-tuning, just by providing like queries and prompts to the model. So in genomics, actually we see some lag in development application of this methodology. So today I think that we are somewhere in like 5 years back in this space, and probably we will have some divergent development here. So today we are working on these, training these DNA language models, which has a very good representation, or we hope that they will have very good representation of DNA sequences to solve all these downstream tasks related to the biology or biomedicine. So the question then, do we have enough data to train these models? Because of course all we know about scaling laws, and we need to scale model size, but we also need to scale data to have good models. And on this graph you can see data used to train different LLM models, and right now actually we already almost depleted all available textual data. We have scrapped all the internet, we have scrapped all code, and now we are facing problem with getting more data to scale our models. But in biology, if we consider all the sequenced genomes we have now, we are in a much better position. First of all, you can see that like during recent years, we have new generation of sequencing techniques which allowed us to sequence much more data. And right now we have like current trends shows that all the DNA data doubles every year and a half. And actually there is a lot of organisms not yet sequenced, a lot of humans not yet sequenced. So we are not, like in the near future, we will not hit this ceiling. And I think that we'll have much more data in the near future on genetic sides. And this is why we have started to develop these GENA models. So it's a very kind and nice model to solve problems of humans. And we adopted a very straightforward approach. We started this project about 3 years ago. We had a very good experience in training bird-like transformer models for natural texts. So we decided to apply it just directly to DNA sequences. So first of all, we applied, we need to split our DNA letters on some chunks to make our models capable to process longer sequences. So we used like standard approach in NLP with a byte pair encoding. This is some scheme which allows you to create optimal encoding for some vocabulary size. As a result, we have a tokenizer with which produces for human genome average token length of about 9 nucleotides, which allows you to process about 4.5 kilobases or 4,500 nucleotides in a 512 tokens input context. Or if you use larger models, you can stretch this context up to 36,000 nucleotides. And we trained these models with a traditional mask language modeling objective, where you have your input sequence. You see that we tokenize it on chunks on our tokens, and then you mask part of your sequence with the mask tokens. And the task of the model is to uncover this mask to predict this mask token. And during learning, this allows models to learn good representation of the input sequences. And after that, after you have pre-trained this model, you should fine-tune it for specific task. And in our case, this might be, as a downstream task, we can have variant effect prediction, which means you predict some effect on the phenotype or splice sites annotation to find how splicing of DNA, like post-processing after translation works, or finding places where transcription factor or these like controlling molecules which controls expression binds to DNA, or detect like taxon of organisms. And after pre-training, we've seen good metrics that our model actually learned something. It started to recover sequences pretty good. But of course, we want to test if the model produces sensible like information about genomes. So as a first test, what we did, we just took different genomes and produced embeddings of different genomes with our model without further fine-tuning. So we just tested how the model can represent different genomes, and then run some traditional clustering algorithms on top of that. And as you can see here, the model like solved this problem like pretty good. So you can see that DNA sequences or genomes from the different taxa. So every point here is just one embedding produced by our model. They cluster nicely, like covering different taxa. So you have fishes here, mammals here, some plants here, birds here. So it's like pretty nice picture. So this is the first like argument that these models this transfer learning should be applicable to DNA sequences. After that, we decided to look in more details what actually model learns during downstream task training. So here we tasked our model to learn to recognize binding sites for these transcription factors, CTCF. So you have this DNA molecule, very long, and CTCF factor, this is a factor which binds in a different parts of the DNA. It's related actually for forming 3D structure of the molecules, of the DNA molecule, because it's like glues together different parts of the DNA. So and by these creating 3D structure, different 3D structures, it controls gene expression. So we can train a model to recognize these stretches of DNA which are where this CTCF binds to, and we know from like traditional bioinformatic tools some motifs how these places should look like, but we are not providing our model with this motif. We're just providing DNA sequences and signal if CTCF binds to this part of DNA or not. And after training, we used feature attribution to find parts of the DNA responsible for this binding. So we wanted to recover which parts of the DNA are important for CTCF binding. And we found actually that this is a part of DNA which has a positive label, that it's CTCF actually binding here. And you can see that attribution method gives like some very high scores to this part of the DNA, which means that these particular parts with the high peaks are related to CTCF binding. And if we look closely, like we have all the all our data set, and when we see which tokens has highest total score, we can rank them, and then we see that actually tokens with a higher score, so which are the most important for predicting CTCF binding, they have some counterparts in this motif recovered by traditional bioinformatics. So which means that our model learned how, like, learn a pattern where CTCF binding proceeds. So it's very nice. But how our model in a zero-shot mode generalized to other species? So we pre-trained our first models on the human data, and then if we fine-tune our human model, fine-tuned for CTCF binding on the recognition of CTCF sites for other species, will it work or not? Can it generalize to other species? And you see it does pretty well. Not for like very distant species, but for closer species it does well, and it can it shows good performance not only for CTCF but for other transcription factors, so other markers as well. So as we know, we have this transfer learning between languages, for example. So if we train model translate between two languages, when we add more data and train this model for other languages, it will give better results if trained from scratch for the given pair of languages. So we also decided to test how our models pre-trained on different species will be able to perform some biological meaningful predictions after fine-tuning for specific tasks. And here we can see the task of promoter prediction. So what is promoter? So the promoter is a part of the genome just before the gene, which is like a regulatory region where different molecules bind to regulate transcription of the gene. So it can initiate, like, if some molecules bind to this promoter region, it can facilitate or inhibit translation of this DNA or expression of this gene. And what we see, so we take 3 different models. One model pre-trained on Drosophila genomes, another model pre-trained on the multispecies data, and another model pre-trained on only human data, and then fine-tune separately these 3 models for the Drosophila promoter prediction. And of course, you can see here that if the model is pre-trained on Drosophila data, it will get better quality for predicting Drosophila promoter binding sites. But also for multi-species data and for human data, it still has pretty good performance, which means that in some applications, so first of all, that all these models learned some grammar rules, some meaning of DNA sequences trained on the totally different species, but also that we can apply model across species to, if we have not pre-trained model for specific species, we still can solve some interesting tasks with the model pre-trained for other species. And recently we have benchmarks published by independent researchers from China, which benchmarked about a few dozens of DNA models for more than 30 tasks related to DNA, RNA, and protein prediction. And we were very happy when we've seen that our models, like different sizes and trained on human data or on multi-species and different sizes, that our models are in top 10 models for all these tasks, and overall our models are the best for DNA. And this is always nice to have good performance on multiple tasks. And also we can start to address questions. Okay, we applied very basic BERT-like model to genomes and we found some pretty good results. So what should be the next? And first question that comes to mind is are scaling laws working for DNA models or not? And here it's from the same study, different models. Here you can see model size, and here average performance score on all data sets. And we see clear scaling law in this range of model sizes, and for the different types of models, transformers, state space models, and so on. And let's consider some particular family of models, nucleotide transformer. So you see, if we start with the 50 million models, then we go to 100 million parameter models, we see better quality, and then we have 5 times more parameters, we get even more quality. But interestingly, after that, if we scale 5 times more, we see no improvement in performance. So the question is, are we hitting the ceiling here or not? Because these are all smaller models, and probably maybe we can see scaling here but not there. And yes, we see some troubling signs from the latest research as well. And this is EVO2 model. Probably it's one of the biggest model we have published today. It's not masked language model, it's autoregressive model, generative model for genomes trained on all available data for 2 different sizes, 7B and like 7 billion parameters and 40 billion parameters. So let's take a look if 4 billion parameters model is better than 7 billion parameters. Here we take this splice variance prediction task where we have mutation and free variants like normal outcomes, splice altering, or in giving exonic or intronic place of this mutation. And you can see here that 40B and 7B models, they are almost the same performance here too. And another task, zero-shot bacterial protein fitness prediction. Again, here, 7 billion parameter models, even like slightly better, but I'm not sure if there is any statistical difference, but no clear advantage of getting larger model, and it's like reproduced for all the tests we have. So the question is, if scaling law is broken, we added much more parameters, but we have the same performance or even worse performance. So what is happening? And actually, I think that probably today we have no answer, no confirmed answer to this question, but maybe we have some, I have some hypothesis why we hit this ceiling and how we can overcome it. And probably it's related to length of the processed sequences. And usually what we found in our tests that longer DNA input means better results. So if you provide more information related to your task, you will get better performance. So here we have like three different models for Drosophila, Arabidopsis, and yeast. So by the way, why we have GENA LM family of models, because we have like different models for multispecies, for these traditional species, for humans, and so you can use them for your tasks. And you can see that consistently, if we compare models which has only 300 base pairs input with 2000 base pairs input during fine-tuning, the longer sequences get your better results. And also review and testing of multiple models for species classification shows that with increasing sequence length, you have better and better and better results. But probably then you have some saturation for the longer sequences. So the question is here, are we limited in this range by the architecture of our current models or some other factors? And to address this challenge of long sequence processing, we started to develop our recurrent transformer architectures based on the very, very simple, similar idea, how we can make our normal transformer to process longer and longer sequences. So what we did, so we take our GENA LM pre-trained model, and then we add to vocabulary new tokens, which we have called memory tokens, and split our long sequence on segments. And we convert, imagine that this is a sum recurrent cell. So you feed your sequence and you feed as an input also these memory tokens. And after they have been processed by the model, we take representation of memory tokens and feed them as an input for the next segment, and then process the next segment. And we train this model on multiple segments and allow gradients to flow back to learn parameters, to fine-tune parameters of a model, to store important information from this segment in memory tokens to be reused on the next segment. So in this case, we can extend our approach to much longer sequences. So after fine-tuning for species classification task, in this setup, we will get much better results. So for example, if we start with 1000 base pairs, we have pretty bad error for species classification. But if you increase 5 times length of your sequence, you decrease this error 100 times. And if you increase sequence length from 50,000 to 200,000, you can add 2.5 times, you can reduce error 2.5 times more. And we see that this is like pretty promising approach to process longer sequences. So let's outline main features of our GENA LM open source genetic models. So first of all, as we've seen, they have a state-of-the-art performance. They on par with specialized model and perform very well compared to other DNA-based models we've seen in independent benchmarks. And we see that these models can do interspecies generalization, and we can fine-tune them for multiple tasks. And with the application of these recurrent architectures, we can process even longer sequences. And I'm very grateful for my colleagues with whom we have worked on this project. And probably you think that this is the end of my talk. Yes? Not. Yes, and this is open source and accessible. But what is the future of genomic AI? Because I think that our GENA LM is already yesterday. Probably today a bit, but we have much more to explore and discover in the future. And this is why I'm so happy to work on this, and I'm so enthusiastic. So first of all, I think, and this is my hypothesis, that why we fail to scale our models, it is due to the nature of genetic data. Because in natural language, we usually have short-term correlations. So we can explain a lot of data just from nearby words or nearby sentences. But this is not the case for genomic data, because in genomic data, we've, first of all, also has these short-range interactions, which is like about 1,000 base pairs. But we also have long-range interactions, about 100,000 or 1 million base pairs. So like these distant parts of the DNA interacts with each other and changes outcomes of like gene expression or all the biological processes inside the cell. And this is why I think that probably just increasing model size, what current models are doing, they're just learning these short-range interactions. And with current sizes, we already learned everything which we can from this part of short-range window. But we are not yet here, we are not understanding this part. And to understand this part, what we need to scale our approaches to full genomes. And how we can do this? Probably we can apply our latest version of recurrent transformer. So here, what we did, I've showed you recurrent memory transformer when we use these memory tokens and feed them inside the model, but the model itself was the same. So our next step, and we tested it in natural language processing tasks, was adding associative memory for every layer of the network to make this memory more resilient to error accumulation. And on natural language processing, this approach worked very well. For example, here, it's a needle in a haystack type test. You see, like, it's for where we tested like normal architectures like Gemini and GPT-4 and some alternatives like Mamba and RAG methods. And you can see that this line, this RMT violet line, it performs well for 1 million tokens, a bit less well for 10 million tokens. But these associative recurrent memory transformer, our new architectures, sets like historical record for the longest sequence to be processed by any neural network, showing that it have more than 80% accuracy for 50 million tokens. Yeah, and this looks pretty promising for applying to DNA. And so we have our associative recurrent memory transformer with 50 million tokens. But if we compare it with the human genome, it have 3 billion base pairs. And even if we tokenize it with 10 nucleotides per token, we still have 300 million tokens, which is 6 times more than our associative recurrent memory transformer can process. And we have all the genomes for all the species, which are pretty different from each other. And this is why I like this area. So we still can produce very meaningful result and solve very challenging problems of scaling our existing architectures beyond what we have for natural language processing in a meaningful way. Imagine that we already trained these full genome scale models, what we can do for science and for medicine with that. So as I've said before, we have, like, if we train it on all species, the model will learn this, like, book of life, and with that we can try to predict with this model. So we will fine-tune it for the different downstream tasks to predict cellular level processes or organ level processes, or even some physiological signals, like how you looks like. As I've said, if we have twins and they are the same for the same genotype, probably in 10 years we will have a model. After you feed in your DNA and you set your age, it will give you a photograph of you at this age. Why not? But of course, the question is, we have these predictions, but we want to understand science. We want to understand what parts of the DNA related to specific biological processes. And I think that our approach opens like a new window. It creates a new tool for biologists to explore all these questions. As I've shown you before with the attribution analysis for this CTCF binding factor, what we can do after we have trained the model to predict outcome of specific biological process, for example, on the cellular level, then what we can do the attribution analysis and find parts of the DNA which are related to this specific process. And then probably do some experiments altering these parts, see how this process will change. So this gives us something, some tool, like a telescope. If you look at the night sky without telescope, you will not see a lot of stars, probably. But if you have telescope, you will see much more. So here we're creating something like a telescope for biology or microscope. And if we can train the model for physiological processes and for cellular processes, and then we can see potentially some overlap. And we can see that some parts of the DNA, they are involved in the cellular processes, and the same parts are important for some organ level processes. Probably we can create a link and our understanding how this cellular level related to the organ level. And we can do the same for the full phenotypes. So just let's play the game. And imagine that we have these models and we need to understand, like we want to address the hypothesis. Is aging the accumulation of errors or it's pre-programmed? We know that today we have no definite answer to this, and how we can explore this with our model. So do you have any suggestions how we can do this? How it will help us to understand if it's pre-programmed or...
Because we have some organisms.
Yes, but we need method to understand if it's in the DNA or not.
Maybe we can train a classifier with the labels, the age as labels, and see if there is a correlation between the genomic data and the age data. Maybe, yeah.
Yeah. So we collect data we collect a database for organisms with the different lifespans, and then train our model given genotype, genome of the species to predict age. Yes, this is the first step. And what we do next? Mm-hmm. Yeah. Yeah, so we can include them in this training data as well. And after that, we can do this attribution analysis. And these two different hypotheses should have a different patterns of attribution. So if we have no specific program which drives aging, that means that our model will have a like blurred attention. We will have no specific parts of genome which determines aging. But on the other hand, if aging some program which turns off at some moment, I would expect to have some specific parts of the genome related to prediction of the age. So this is a nice example how we can use these DNA models, future DNA models, as a tool for addressing fundamental biological questions. So here is my roadmap to decoding life with AI. So AI can learn genetic code, and this genetic code is a programming language of life. And we should use genome-scale DNA language models for that. And these models will enable prediction and interpretation of molecular, cellular, and organismal processes directly from genome sequences. And as more genomes are sequenced, we will decipher processes across all known organisms. So I think that the more data we will have, we will gradually gather more and more language and deeper understanding of all life on Earth. And this will empower us to answer biology's most fundamental questions and, of course, improve human health, enabling personalized medicine, early disease prediction, and novel therapeutics. Thank you for your attention.
Thank you for a very interesting talk. And I will start with the first question. So I'm curious if it is possible to use the single-cell RNA data to fine-tune the model for prediction of the cell type.
Yeah, this is actually what we are doing right now, and it's pretty hard task. So let me explain more to other people. So what we have today, we have a lot of data where we have a single-cell RNA sequences which tells us how different genes are expressed in particular cell type. Not only genes, but also these regulatory parts. And these patterns of expressions, they are unique to different cell types. And what we have today, it's like this expression data, which says how many times specific sequence was read from the DNA. And we also have a textual description, like what is cell type, what is a species, what is a age, and some other data. And actually, the idea is that we can train multimodal model, like taking this foundational model, and then add textual description as a signal which tells the model how particular part of the DNA should be translated into the RNA data. And yeah, we are working on this because this is very important for understanding biological processes. So like, given this description, what parts of the DNA will be like transcribed, and what are the kind of like also controlling signals to change this expression.
And how you handle batch effect?
Yeah, it's mainly during pre-processing. Like we want to normalize data, removing all this batching effect and so on. But of course, we started from first experiments, we're just trying to push all the data as we have and seeing what is happening. But model starts to learn this batching, like average levels of expression for different batches. So we started to remove it from the data.
Thank you. Do we have other questions?
Thank you for your impressive talk. I want to ask about some questions about the data. So as you said, you have an old genome. How do you batch your data for the feeding the like a large model? So, are you when you are choosing what is the size of the tokens that you are using? So, do you trying to choose from the genome, like different parts of the genome, like parts that are answering about the genome, or how do you curate your data?
So as I've said, that our initial approach was to use byte pair encoding tokenization. So we took a human genome, and then set a vocabulary size, probably 30,000 tokens, and then use BPA to create these tokens. And then during training, we have a model with a 512 input size, BERT-like model. So we just have the chunks of these size and batched them, like shuffling all the.
So they are random, and also while you are training, you can use like a human genome or different mammals, and or all shuffling them.
Yes. So we have like multiple models. We have one model trained only on human genome, another model trained on like 7 genomes from different species, and also we have specific models for Arabidopsis, yeast, and Drosophila, which is trained only on Drosophila genome.
Do you use like repetition of the genome in your data? Like you are trying to repeat the chromosome parts, or the translation parts, or you are just one genome?
Yeah, it's like we treat all parts of genome as equal, so we're not upsampling anything. So if we like going one epoch, another epoch, so we're just shuffling data from the same genome uniformly.
Okay, and one more question: is the connections of your, as you said, associative blocks are randomly, or they just are you choosing that connections randomly?
Which one?
The slide about the associative blocks.
Here,
yeah. One associative block to the other these connections random, or how do you choose what block to connect?
Yeah, the model should learn this. It's just some general mechanism. So what is happening, this associative block, it's some memory, and after every segment, so you like, here segments are, it's continuation of the same sequence. So they are not segment 1, in segment 2 they are not randomly sampled. And here, these associative blocks, in its activations, it stores some information transferred from these memory blocks. And then we copy this information, then update it from the memory.
Thank you very much.
Thank you for the talk. I had a question about your tokenization process, because I suppose you use the probabilistic method, like in languages, to find the tokens. But in DNA, probably there is no logical divide in between the genomes. Like this one is the start of the word, is the end of the word. And when you look for like tasks that are targeting locations in the gene, can the tokenization hinder this process, where the actual location is in the middle of your token, so you don't have the actual location?
Yeah. This is a challenging. If your task requires, for example, you want to segment your DNA sequence, then you need to have a precision of 1 letter. And in this case, we use 2 approaches. One approach, we just tokenize with 4 tokens, because in our vocabulary we have tokens for 1 letter. It's one approach. And another approach, we use our initial model as an encoder, which produces vector for tokens. And after that, we take these vectors and we take single nucleotide embeddings, so the same sequence on this single nucleotide resolution, and combine like embeddings produced by our models with these new single nucleotide embeddings. And in this case, we have a single nucleotide resolution, and we feed it in some upper layers, which we fine-tune. So like we increase resolution for the higher levels, and then out of these embeddings, we predict this like a solution for this task.
Thank you.
You're welcome.
Thank you for the talk. So I have a question about aging. So you said that there can be two situations where we have attention maps very blurry, and we can treat that as we don't have pre-programmed aging, and the second situation where we have very tight and precise attention maps. But do we have a threshold to understand? Just about the threshold when we say that it is pre-programmed or not.
I don't know. We haven't yet. I think that when we will start to work on this problem, probably we can see if we will see any difference. Is it statistically different? We have statistical difference between this region or not, or we need to have some other control experiments to train model for predict some other feature and see how it will work.
Thank you.
Well, thank you for a very interesting talk. So there is this DNA Foundation model EVO, and EVO 2 was also released recently. What is the comparison between GENA and EVO? For which tasks, which one overperforms the other one?
We have no direct comparison with EVO because GENA was released before EVO and the EVO 2, so we have no head-to-head comparison. And unfortunately, EVO was not included in this large-scale benchmarking, which I've shown. But probably you remember that I've showed you this scaling thing here. This for EVO, and it shows that it doesn't scale. But actually, it's I think a very interesting model because it has potential to process 1 million tokens, 1 million base pairs, because it's not token level, it's a base pairs level, and it's generative model, and we have some comparison with the previous versions, which was called Hyena DNA for these species classification task. And for species classification task, our model for 200K was better than their model for 1 million. But the difference is very tiny.
Okay. Do we have other questions?
Like, if you speak about aging, I mean, there are like mutations that happen. Like, how do you handle that in the environment? Also, like cells during their lifetime will have different genomes. So you know, like the cancer is one of the reason of the like organism dying.
Yeah, I don't know how it is directly related to aging, but I think that in terms of training, pre-training, you will have. I understood. So you mean that during lifetime mutations accumulate, and these can cause aging. Yeah, and then it fits accumulating error hypothesis. So then it's the same as error accumulation. So we treat genomes without mutations, but if mutations accumulate, which means that they are uniformly like altering state of our genome. Sorry. Yeah, then it's this thing. No. And this is, I gave this as an example. This is not we are working on right now or plan to work in like 2-5 years. We are not there. I think it's like, it can be addressed in 7-10 years ahead.
Okay. Thank you very much for the interesting talk.
Thank you for invitation.
Yeah.
Thank you for your attention.
Yeah.